All right. Good afternoon, everyone. Thank you for joining this lecture related to pharmacology of autonomic nervous system. And first, I would like to ask you to type in the chat about quality of this streaming, about quality of this translation, because it's my first experience in YouTube Live. So I would like to ask you how you can hear, everything is clear, and if you are able to view this translation in HD quality, like 1080p. So please tell me, I would appreciate your feedback. Now I would like to say a few words about plans for today. So today and tomorrow we will talk about pharmacology of autonomic nervous system. We will cover general aspects of autonomic nervous system pharmacology. We will talk about receptors. We will talk about second messengers. We will talk about localization of these receptors and effects they cause. And today and tomorrow we will cover organs based on what kind of receptors are present in these organs. I hope this approach will be useful for you, and let's start. So first, let's start from classification of nervous system in order to understand where the autonomic nervous system is. So nervous system classification. As you know, nervous system is classified into the central nervous system, CNS, and the central nervous system includes brain and spinal cord, and we have peripheral nervous system. Peripheral nervous system. Peripheral nervous system subdivided into two branches. The first one is the afferent division, and second one efferent division. Afferent division sending signals directly to central nervous system, and it is kind of sensory input. Okay, thank you for your feedback. Okay, video and sound quality good. So afferent division sending signals to the central nervous system, and efferent division subdivided again into two parts. The first one is a somatic one, or it's also can be mentioned as a somatic nervous system, and we have autonomic nervous system here. What does it mean, somatic nervous system? Somatic nervous system regulates skeletal muscles, yeah, and regulates like it's under voluntary control. I'm writing here voluntary control, and innervates skeletal muscles. Autonomic nervous system, as you know, subdivided into 3 parts. Yeah, sometimes you can meet 2 parts, but really we have 3 parts. The first one is the sympathetic nervous system. Second one is the parasympathetic nervous system, and the third one, the enteric nervous system. Just would like to mention that sympathetic nervous system responsible for fight and flight response, and the parasympathetic nervous system responsible for rest and digest response. But what I would like to say in terms of pharmacology and in terms of physiology, I would like to mention major neurotransmitters in each division of autonomic nervous system. So as we're talking about sympathetic nervous system, we should say that major neurotransmitters are epinephrine and norepinephrine. Epinephrine and norepinephrine. Sometimes you can meet another names of these neurotransmitters, and sometimes it can be mentioned as adrenaline, epinephrine, and norepinephrine as a noradrenaline. For parasympathetic nervous system, major neurotransmitter is acetylcholine. And let's say drug classes, that act on the sympathetic nervous system and parasympathetic nervous system. Drugs that produce effects similar to epinephrine and norepinephrine, called adrenergic agonists. Or you can meet another term, adrenomimetics. Adrenomimetics because they mimic action of adrenaline, or epinephrine. Or you can meet adrenergic drugs. So basically, it's the same medications, the same drugs, but with different names. Drugs that prevent action of epinephrine and norepinephrine, called adrenoblockers. Adrenoblockers, or they called adrenergic antagonists, or anti-adrenergic drugs. Anti-adrenergic drugs. The same situation with cholinergic medications. Drugs that mimic action of acetylcholine, called cholinomimetics. Or cholinergic agonists, or generally we can say that they are cholinergic drugs. Drugs that block action of acetylcholine, called cholinoblockers, or anti-cholinergic drugs, or cholinergic antagonists, or another term, cholinolytics. The same term can be applicable for sympathetic nervous system, but these medications called sympatholytics. Sympatholytics. So they block, they prevent action of basically, let's say, epinephrine and norepinephrine on the receptors. Okay. Next, let's say a few words about localization. Localization of sympathetic and parasympathetic nervous system centers. Localization. Let's make kind of table. Yeah. Let's put here sympathetic nervous system, and let's put here parasympathetic nervous system. First, we should see origin. Sympathetic nervous system has thoracolumbar origin, right? So it is a TH1 to L2, L3 segments, and the parasympathetic nervous system has craniosacral localization. Craniosacral, we have cranial nerves. Yeah, which one? The third, oculomotor nerve. It is nerve number 7, facial nerve, glossopharyngeal nerve number 9, and nervus vagus 10. About glossopharyngeal nerve and nervus vagus as the most important, we will talk tomorrow when we will discuss baroreceptor reflex, and when we will talk about blood pressure regulation and heart. Yeah, again, distribution. What does it mean, this distribution? You can meet information that sympathetic nervous system has a wide distribution, and parasympathetic nervous system has limited distribution. But what does that mean really? What is that? Yeah. Wide distribution. You know that adrenal medulla releases epinephrine and norepinephrine. And this adrenal medulla releases epinephrine and norepinephrine into the bloodstream. And as you should understand, that blood go into all organs and tissues. Yeah. So these catecholamines, epinephrine and norepinephrine, they are coming to all cells, to all tissues. This is called wide distribution. Yeah. For parasympathetic nervous system, distribution is limited. Why it is limited? Because it is limited to nerve terminals related to parasympathetic nervous system, limited to the nerves. Yeah. Next one, ganglia. Most ganglions of sympathetic nervous system are present in paravertebral chain, so they are away from organs. Away from organs, and ganglions of parasympathetic nervous system close to organs. And sometimes they might be mentioned as an intramural. Ganglions, because they present inside the organ wall. Fibers. Now we will talk about that. So we have short preganglionic and long postganglionic fibers. And vice versa for parasympathetic nervous system. For parasympathetic nervous system, we have a long preganglionic and short postganglionic fibers. Neurotransmitters. We already said that major neurotransmitter for sympathetic nervous system is norepinephrine, because norepinephrine, it is a major neurotransmitter that released from synaptic terminals, from sympathetic nervous system fibers. It is norepinephrine. Epinephrine released, as we said, from adrenal medulla, from here. And as we're talking about adrenal medulla releases 80% of epinephrine and 20% of norepinephrine. So major neurotransmitter for sympathetic nervous system is norepinephrine, but epinephrine also. And major neurotransmitter for parasympathetic nervous system is acetylcholine. Now we should say a few words about, I'll scroll it a little bit up. Yeah. Now we should say a few words about enzymes. Enzymes that are responsible for degradation of these neurotransmitters. Enzymes. Most likely you know that for acetylcholine we have acetylcholine esterase. This acetylcholine esterase are predominantly present in synaptic cleft and postsynaptic membrane, and it is responsible for degradation or for breakdown of acetylcholine molecules into two small parts. It's the acetyl part and choline part, and choline part undergo reuptake. But also we have butyrylcholine esterase. Butyrylcholine esterase. This butyrylcholine esterase present in blood plasma, yeah, and it degrades also acetylcholine, but responsible also for degradation of local anesthetics and depolarizing skeletal muscle relaxants that called succinylcholine, and genetical abnormalities related to deficiency of this butyrylcholine esterase manifest in long action of succinylcholine. If it usually acts around 5 to 10 minutes, in case of deficiency, effects of succinylcholine may last hours. And this is important to know. What about norepinephrine and epinephrine? Two enzymes we have, COMT and MAO, catechol O-methyltransferase, and monoamine oxidase. Monoamine oxidase present inside synapse, and catechol O-methyltransferase present outside. Do not think, please, that these enzymes for 100% are responsible for degradation of, not for degradation, but for termination of action of these neurotransmitters. The major mechanism of termination of action for adrenergic synapses, the reuptake. So around 80% of norepinephrine that present in synaptic cleft going back with norepinephrine reuptake transporter, and synapse use it again. But the major mechanism of termination of action in cholinergic synapses is a hydrolysis by acetylcholine esterase. I will try to make, I'm just looking on your comments, I will try to make this video available on my YouTube channel, and maybe I'll split it into the small sections, so it will be much more easier to watch and review, these small parts than watch a whole long video. Okay, now let's talk about, so guys, if you have any questions, please type these questions in the chat, and as I will finish section, I will try to answer your questions. Yeah. Okay. Now let's talk about receptors and neurotransmitters. Receptors and neurotransmitters. As we said, we have sympathetic nervous system and parasympathetic nervous system. We have different types of receptors for them. Let's see, in parasympathetic nervous system, we have cholinergic receptors, and in sympathetic nervous system, we have adrenergic receptors. What kind of cholinergic receptors we know? We have, we know two types of cholinergic receptors. The first one is a nicotinic receptors, and second one muscarinic receptors. Why they nicotinic and why they muscarinic, and how do they work? They nicotinic receptors because they sensitive to nicotine, yeah, and muscarinic because they sensitive to muscarine as a mushroom alkaloid. But both these receptors are sensitive to acetylcholine, and acetylcholine is a endogenous ligand for them. Nicotinic receptors are subdivided, let's say, into two types or two subtypes that we really need to know. In real situations, much more subtypes, but I would like to put two types only here, two subtypes, an N subtype and an M subtype. What does it mean, N? N means nerve, yeah, and they present between neurons, between neuronal cells, and M related to muscle. So it is a skeletal, they present on skeletal muscles, and it is a nerve-muscle junction, nerve-muscle junctions. Both these types of nicotinic receptors are ion channels. And if we would like to be more precise, they are ligand-gated ion channels. Ligand-gated ion channels. Let's illustrate that. So we have acetylcholine molecule attached to this ion channel, and ion channel opens and sodium ions are going inside the cell, and as a result we have depolarization, yeah, and maybe action potential generation. What is interesting? Interesting that these ligand-gated ion channels can be present in three different states. What kind of states? The first state is a closed but ready to open. We have two gates in every channel, M gate and H gate. So we have opened H gate and M gate is closed. This state like initial one. As a ligand attaches to this ion channel opens. So we have opening in H gate and we have opening in M gate, and this second state called open. In this situation, in this condition, sodium ions are going inside and depolarize the cell. And third state called inactive. Inactive, it is a situation when M gate is open and H gate is closed. What is important? Important that transition occurs only from inactive to ready to open state, and never to open state. So inactive channels are unable to become opened. Yeah, they should go to closed but ready to open state, and after that they can be opened. Muscarinic receptors. Muscarinic receptors subdivided into 5 subgroups, and it's very easy to memorize them, as they called M1, M2, M3, M4, and guess the last one, yes, M5. M5. They are metabotropic receptors, and they called GPCR receptors or G protein couplet receptors. Yeah, G protein couplet receptors. Now we will talk about that. G protein couplet receptors. What about adrenergic receptors? What about adrenergic receptors? Adrenergic receptors are subdivided into two big groups. The first one, the alpha receptors, and second one, beta adrenergic receptors. And we have two subgroups in alpha. It's alpha 1 and alpha 2, and three types of beta adrenergic receptors. Again, beta 1, beta 2, and beta 3 receptors. Really, beta 3 receptors responsible for lipolysis in adipose tissue, lipolysis. And another place where they are present are the skeletal muscles, and they are responsible for thermogenesis in skeletal muscles. Thermogenesis in skeletal muscles. Here is a very important concept I would like to mention about ligands and receptors. We said that major neurotransmitters for sympathetic nervous system is epinephrine and norepinephrine. Epinephrine produces action on alpha 1, alpha 2, beta 1, beta 2, beta 3 receptors. And norepinephrine produces action on alpha 1, alpha 2, and beta 1 adrenergic receptors. So where is the difference? Difference in the action on beta 2 adrenergic receptors for epinephrine, if we will contrast epinephrine with norepinephrine. And this is important. This is important for vasorelaxation and important for relaxation of smooth muscles, because through beta 2 adrenergic receptors we have mediation of this process. So as dosage of epinephrine rises, it produces action on alpha 1 receptors. Yep, it produces vasopressor action. In low dose, epinephrine acts predominantly on beta 2 adrenergic receptors, and as dosage rises, epinephrine produces action on alpha 1 adrenergic receptors. Of course, beta 1 and alpha 1. Yeah, but action on alpha 1 receptors predominate as dosage rises. For norepinephrine, we should keep in mind that norepinephrine acts more on alpha 1 adrenergic receptors. And it produces vasopressor action. Okay. So now let's contrast second messengers for these receptors. Second messengers for these receptors. Let's put M1, M2, M3, the 7 times transmembrane receptors, yeah, M4, M5 receptors. After that, alpha 1, alpha 2, beta 1, beta 2, beta 3. Sometimes I draw 7 times, sometimes not 7 times, just to illustrate to you, yeah? So, acetylcholine acts on M1, M2, M3, M4, M5 receptors, and let's say epinephrine, norepinephrine, acts on alpha 1, alpha 2, beta 1, beta 2, and beta 3 receptors. All these receptors are G protein couplet receptors. M1 couplet with GQ, M2 with GI, M3 GQ, M4 GI, M5 GQ, alpha 1 couplet with GQ receptor protein, alpha 2 GI, and beta 1, beta 2, and beta 3 receptors couplet with GS proteins. What does that mean? It means that for M1 receptors, or M1 receptors act through inositol triphosphate diacylglycerol and calcium cascade. GI adenylate cyclase and CMP. Again, GQ inositol triphosphate diacylglycerol, yeah. GQ, again GQ, IP3 diacylglycerol. This is important to memorize all these second messengers. Here I'm writing CMP, and again CMP, MP, and GS, again cyclic adenosine monophosphate cascade, yeah. But what does it mean, and how to memorize these things? Look, M1 receptors, M3 and M5 and alpha 1 couplet with calcium inositol triphosphate diacylglycerol cascade, yeah. So most likely a result of excitation of these receptors, or a result of binding of ligands with these receptors, will be either contraction of muscles, yeah, smooth muscles, either contraction or secretion of something, because secretion is also active process that required calcium ions. M2, M4 and alpha 2 receptors couplet with GI or inhibitory G protein, yeah. GI is an inhibitory protein, and this inhibitory G protein inhibits adenylate cyclase, and as a result of inhibition of adenylate cyclase, we have decreasing in cyclic adenosine monophosphate level. And beta 1, beta 2, and beta 3 receptors coupled with GS or stimulatory G protein, they stimulate activity of cyclic adenosine, stimulate activity of adenylate cyclase and increase amount of cyclic adenosine monophosphate. So let's do next thing. Once again, let's put all these receptors in the line, and let's put here M1, M3, M5, and alpha 1 adrenergic receptors. All these receptors are coupled with GQ protein, right? But we should remember that acetylcholine is a ligand for M1, M3, M5, and epinephrine and norepinephrine are ligands for alpha 1 adrenergic receptors. M2, M4, and alpha 2 receptors coupled with GI or G inhibitory protein. Yeah, and again, acetylcholine is a ligand for M2 and M4, and epinephrine and norepinephrine is a ligand for alpha 2 adrenergic receptors. And beta 1, beta 2, and beta 3 receptors have common ligand epinephrine, norepinephrine, but they coupled with GS, with G stimulatory protein, and both these proteins, they regulate activity of adenylate cyclase. The only difference is excitation of M2, M4, and alpha 2 receptors causes inhibition of adenylate cyclase, and excitation of beta 1, beta 2, and beta 3 receptors causes stimulation of adenylate cyclase. Yeah, so we can put minus here, it means inhibition, and plus here as a stimulation. And adenylate cyclase responsible for conversion of ATP to cyclic adenosine monophosphate, and as a result we have different cellular effects. Yeah, different effects. What is interesting? Stimulation of M2, M4, and alpha 2 receptors inhibits activity of adenylate cyclase, but blockade of these receptors will have opposite effect, will stimulate activity of adenylate cyclase or enhance activity, yeah, or decreases inhibition, and vice versa. Excitation of beta 1, beta 2, beta 3 receptors stimulates adenylate cyclase activity, and blockade of these receptors will inhibit adenylate cyclase activity. Yeah, adenylate cyclase activity. Now let's see a few words about inositol triphosphate, IP3, diacyl glycerol cascade. So we have cells, right, and it is a cellular membrane, outside, inside, and this cell we have GPCR receptor, and as we have ligand, and this ligand attaches to this receptor, this causes conformational changes of this receptor. Yeah, inside the cell, this receptor coupled with so-called G protein, and this G protein has three domains: alpha, gamma, and beta domain. Alpha domain also bounded with GDP, guanosine diphosphate, and as we have binding of a ligand, it causes conformational changes, and it causes detachment of alpha G protein and substitution of GDP to GTP, guanosine triphosphate. Yeah, so it becomes phosphorylated, and as it becomes phosphorylated, it activates another enzyme that present in the cell membrane. It's called phospholipase C. Phospholipase C, and phospholipase C cleaves phosphatidyl inositol biphosphate, into two parts. The first part is a diacylglycerol, DAG, and another part is a inositol triphosphate. They have a little bit different properties, DAG and inositol triphosphate. DAG still hydrophobic, and it's still be present inside the bilipid layer of plasma membrane. And inositol triphosphate is a polar molecule, so it very well dissociates and very well diffuses, yeah, in the water. So what is the next? Next, we have endoplasmic reticulum, and in this endoplasmic reticulum, we have IP3 sensitive calcium channels. So inositol triphosphate binds to IP3 sensitive calcium channels, activate them, and causes releasing of calcium to intracellular space. Calcium ions bind to calmodulin, and produce calcium calmodulin complex. Calcium calmodulin complex. And this complex important for stimulation of different proteins, and different protein kinases. So we have stimulation of protein kinases, and one of these protein kinases is a protein kinase C. So we have stimulation of protein kinase C, to which diacylglycerol is bounded also. And as a result, we have cellular effect. What about GIGS pathway. Adenylate cyclase pathway. Again, we have membrane, yeah. We have G protein couplet receptor with alpha, beta, and gamma subunits, and GDP molecule is bounded to alpha subunit. As we have binding of a ligand with GPCR receptor, we have the attachment of alpha subunit and substitution of GDP to GTP, and activated alpha subunit activates adenylate cyclase. This adenylate cyclase is responsible for conversion of ATP to cyclic adenosine monophosphate, and CMP can activate different protein kinases again. Activation of different protein kinases, especially protein kinase A. This causes phosphorylation of amino acids or proteins, yeah, proteins, and as a result, activation of different molecules. Another way is that cyclic adenosine monophosphate is able to activate different important molecules like transcription factors. Activation of transcription factors and this regulates gene expression, and gene expression finally regulate activation and production of different enzymes. Termination of action. As we have alpha subunit bounded with GTP, this GTP slowly hydrolyzed from alpha subunit. So with help of water molecules, yeah, a lot of water molecules present inside the cell, we have hydrolysis. And we have again alpha subunit bounded with GDP molecule and phosphate. This part or this complex or GPCR receptor can be regulated by two ways. The first way, as we have binding of the ligand to this GPCR receptor, and ligand has low affinity and very quickly dissociates from GPCR receptor, this terminates action of this ligand. But if ligand binds for a little bit longer, we have another mechanism with help of beta-arrestin kinases and ATP beta-arrestin and kinases. Yep. Sorry, not with beta arrestin, beta adrenergic receptor kinases. We have phosphorylation of beta receptor, let's say, and after that we have attachment of beta arrestin. And this causes internalization of the internalization of the receptor. Internalization. And as a result, less amount of receptors are present on the cell membrane. Okay. Next, we should talk about cholinergic synapse. We should talk about cholinergic synapse. So let's start from sodium choline cotransporter. With sodium choline cotransporter. This sodium choline cotransporter pumps choline molecules inside the cholinergic synapse, and choline molecules present inside it. Mitochondria produce acetyl coenzyme A. And with help of enzyme that called choline acetyl transferase, we have synthesis of acetylcholine molecules. These acetylcholine molecules should be packed inside synaptic vesicles. And in these vesicles we have a specific transporter that called vesicle-associated transporter. This vesicle-associated transporter pumps acetylcholine inside the vesicles, and pumping out hydrogen ions. It creates negative charge inside the vesicle, so acetylcholine better attached to the walls of these vesicles. Yeah? Experimental medication that called group of medications that called hemicholiniums are block this choline cotransporter, sodium choline cotransporter, and prevent reuptake of choline inside the synapse. Another group of medications, another medication called vesomicol, it's also experimental medication, blocks vesicle-associated transporter and also prevents accumulation of acetylcholine inside inside the vesicle. What is interesting? Interesting that this vesicle has specific proteins on its surface. These proteins are important for docking as this vesicle going to presynaptic membrane. Yeah, it should attach to this presynaptic membrane, and after attachment we have so-called docking and fusion, yeah, and after that we have releasing of acetylcholine molecules into the synaptic cleft and further diffusion of these molecules. So these proteins are very important, and they called SNAP protein, VMP, and syntaxin. Now we will talk about them. They were important for botulotoxin action, for toxin of Clostridium botulinum, as Clostridium botulinum produces a toxin that called botulotoxin, and botulotoxin blocks these proteins, and it prevents docking of vesicles with acetylcholine. Yeah, so botulotoxin belongs to neurotoxin. So, okay, what do we have? After accumulation of acetylcholine molecules into the vesicle, we have docking, fusion, and a releasing of acetylcholine molecules into the synaptic cleft. In synaptic cleft, we have receptors. Yeah, it depends from the tissue. We may have nicotinic receptors, NN or NM. It depends from the type of synapse. And we can have M1, M2, M3, M4, and M5 receptors. So acetylcholine interacts with these receptors, again dependent from the tissue, and produces effect. Also, this membrane, postsynaptic membrane, we have enzyme that called acetylcholine esterase. And acetylcholine esterase cleaves acetylcholine molecule into two parts, the choline part and acetyl part. So choline going back and reuptaken back for a resynthesis of neurotransmitters. So again, major mechanism of termination of action in cholinergic synapses is a hydrolysis by acetylcholine esterase. And what kind of drugs we have here? We have medications that block this acetylcholine esterase, yeah, or they called anti-acetylcholine esterase drugs or inhibitors. Acetylcholine esterase inhibitors. What kind of medications? We have donepezil, let's say, medication for Alzheimer's disease treatment. We have phosphororganic substances. We have insecticides. And warfare gases like azarin, like azaman, weak gases, and others. Yeah. So all these drugs will block acetylcholine esterase, and you should understand, as we are blocking acetylcholine esterase, we prolong the half-life of acetylcholine in the synaptic cleft, and acetylcholine produces greater stimulation of postsynaptic receptors, postsynaptic receptors. A few words about adrenergic synapse. Adrenergic synapse. Adrenergic synapse releases norepinephrine. Releases norepinephrine and amino acid, useful amino acid for synthesis of norepinephrine is called tyrosine. So we have sodium tyrosine co-transporter, or sodium dependent carrier, sodium dependent carrier, and tyrosine appears inside the neuron with help of tyrosine hydroxylase. With tyrosine hydroxylase, we have synthesis of DOPA, and DOPA converted to dopamine. With help of decarboxylase, or aromatic amino acid decarboxylase. So dopamine going inside the vesicles, but here we have vesicle associated monoamine transporter. This vesicle associated monoamine transporter pumps dopamine inside, and inside we have conversion of dopamine to norepinephrine, and also vesicle associated monoamine transporter pumps out hydrogen ions. Synthesis of DOPA is a rate limiting step for adrenergic synapses with tyrosine hydroxylase. So we have medication that's called metyrosine, and this metyrosine blocks tyrosine hydroxylase and prevents synthesis of norepinephrine. Another medication that belongs to sympathetic group called reserpine. And reserpine blocks vesicle associated monoamine transporter. In this vesicle we also have proteins, SNAP protein, this like and in cholinergic synapse. But medications like Reticulum and guanethidine block fusion also. So after docking and fusion, we have releasing of norepinephrine into the synaptic cleft, and the norepinephrine interacts with alpha 1, beta 1, beta 2, let's say, and beta 3 receptors, that present there, and as a result we have effect. But major mechanism termination of action is reuptake. So norepinephrine molecules reuptake it back, and we have a specific transporter, norepinephrine reuptake transporter. And with help of norepinephrine reuptake transporter, we have reuptake of norepinephrine molecules, and again these norepinephrine molecules are packed into the vesicles and reused again. What about alpha-2 adrenergic receptors? Alpha-2 adrenergic receptors present presynaptically, and they play important role in negative feedback. This negative feedback is a kind of auto-regulation for synapse, yeah, and norepinephrine acts with alpha-2 adrenergic receptors, and alpha-2 adrenergic receptors, we remember that they coupled with G inhibitory proteins, and they regulate activity of potassium channels. We have opening of potassium channels, we have hyperpolarization, and a less amount of norepinephrine released into the synaptic cleft. What else? What kind of medications we have? Alpha-2 adrenergic agonists. Most likely you heard about them as a clonidine and methyldopa. And medications that act on norepinephrine reuptake transporter are cocaine, amphetamines, and tricyclic antidepressants. Mechanism of termination of action. First is the reuptake. After that, we have catechol-O-methyl transferase, and we have monoamine oxidase inside. Catechol-O-methyl transferase and monoamine oxidase inside. Nice. Now, let's finally talk about autonomic nervous system. Yeah, as we finished all basic things, and now we are able to operate with different terms when we will talk about neurotransmission in autonomic nervous system. So now we will discuss general approaches to neurotransmission in autonomic nervous system. As we said, we have two divisions. We have parasympathetic nervous system, and we have sympathetic nervous system. Preganglionic fibers. Of sympathetic nervous system and parasympathetic nervous system release acetylcholine, because we have ganglion, yeah, and we have postganglionic fibers. So this is a ganglion. There's a place where we have communication between neurons. This is a ganglion. So inside ganglions, preganglionic fibers of sympathetic nervous system and preganglionic fibers of parasympathetic nervous system release acetylcholine. And acetylcholine interacts with an subtype of receptors on neuronal cells. Neuron, yeah, neuronal subtype of receptors. Also, we have acetylcholine esterase here, and this acetylcholine esterase is responsible for acetylcholine breakdown and degradation. Postganglionic fibers. Postganglionic fibers of parasympathetic nervous system release acetylcholine also. And this acetylcholine degrades with help of acetylcholine esterase, and acetylcholine interacts with M1, M2, M3, M4, M5 receptors. It would be nice if you will know just 3 of them, M1, M2, M3. It will be enough. So, once again, preganglionic fibers of parasympathetic nervous system release acetylcholine. This acetylcholine interacts with an N subtype of cholinergic receptors, and postganglionic fibers of parasympathetic nervous system release acetylcholine also, and this acetylcholine interacts with M1, M2, M3, M4, M5 receptors. As we're talking about sympathetic nervous system, preganglionic fibers of sympathetic nervous system also release acetylcholine. Acetylcholine interacts with an N type of receptors, degrades by acetylcholine esterase, and postganglionic fibers of sympathetic nervous system release norepinephrine. And this norepinephrine interacts with alpha 1, beta 1, beta 2, beta 3 receptors, and alpha 2 receptors located presynaptically. Degradation with help of monoamine oxidase plus catechol or methyltransferase and reuptake. What is interesting? Interesting that we have few exceptions for sympathetic nervous system. Let's have a look. These nerve terminals that release acetylcholine called cholinergic. So preganglionic fibers of parasympathetic nervous system are cholinergic. Preganglionic fibers of sympathetic nervous system are cholinergic. And postganglionic fibers of parasympathetic nervous system also cholinergic, because they release acetylcholine. Postganglionic fibers of sympathetic nervous system are adrenergic. Postganglionic fibers of sympathetic nervous system that innervate sweat glands or thermoregulatory sweat glands also release acetylcholine. And this acetylcholine degrades by acetylcholine esterase. So we should remember, that postganglionic fibers of sympathetic nervous system release acetylcholine in case of innervation of thermoregulatory sweat glands. Preganglionic fibers of sympathetic nervous system that innervate adrenal medulla also release acetylcholine. As a result of rising in sympathetic nervous system tone, preganglionic fibers release acetylcholine to adrenal medulla. Acetylcholine interacts with NN type of receptors, and in response to that, adrenal medulla releases epinephrine and norepinephrine into the circulation. Again, 80% of epinephrine and 20% of norepinephrine. And most likely you heard that in the lungs we have beta 2 receptors. Yeah. When we're talking about lungs, of course, we're talking about smooth muscle cells in the lungs, and most likely you heard that. You heard about that. But here is my question: how does these beta 2 adrenergic receptors in the lungs are innervated if we say that norepinephrine as a major neurotransmitter and norepinephrine does not interact with beta 2 adrenergic receptors? The answer is that epinephrine from systemic circulation produces action on beta 2 adrenergic receptors in the lungs and causes bronchodilation. Sometimes you can meet information that muscle erector pilia innervated by cholinergic fibers, but in muscle erector pilia innervated by cholinergic fibers and coming from sympathetic nervous system that release acetylcholine, and also innervated by adrenergic fibers, postganglionic adrenergic fibers that release norepinephrine. So I would like to put muscle erector pilia here, muscle erector pilia, and would like to show two types of receptors, the alpha 1 receptors and M3 type of receptors. Yeah, and more norepinephrine are responsible for innervation of muscle erector than acetylcholine. Just keep in mind that. Okay. Next. Let's say a few words about medications. Yeah. Few words about medication. Again, parasympathetic nervous system. We have preganglionic fibers. We have ganglion, postganglionic fiber, and sympathetic nervous system. Preganglionic fiber, ganglion, and postganglionic fiber. As we have ganglion, we have a specific group of medications that called ganglia blockers. Ganglia blockers. Ganglia blockers block transmission in autonomic ganglia. They block transmission in ganglions of parasympathetic and sympathetic nervous system. What kind of medications? Let's say hexamethonium. And as a result of action of this medication, we have peripheral denervation. Peripheral denervation. What does it mean? Peripheral denervation. It means that ganglia blockers prevent transmission in ganglions of autonomic nervous system. So they block NN type of receptors, and they prevent transmission of information in ganglions of sympathetic and parasympathetic nervous system. As a result, preganglionic fibers are sending impulses, but these impulses are not transmitted to postganglionic fibers, and postganglionic fibers are silent in this situation. So we have denervation, peripheral denervation. Would like to contrast and not to confuse you with the somatic nervous system. Somatic nervous system responsible for skeletal muscle innervation. So we have skeletal muscle here, and in skeletal muscle we have an M subtype of receptors, nicotinic receptors. We do not have ganglions, you see, no ganglions here. An M type of receptors, and ganglia blockers are not working on an M type of receptors. Instead of that, we have a group of medication that called skeletal muscle relaxants, with depolarizing and non-depolarizing type of action, like tubacurarine, there's a prototypic agent, rocuronium and other, or succinylcholine with depolarizing type of action. So don't be confused. Next one. We see that sympathetic nerve fibers, preganglionic sympathetic nerve fibers, release acetylcholine in case of innervation of adrenal medulla with epinephrine and norepinephrine. So ganglia blockers also act here, as we have an subtype of receptors, and we should consider that like a ganglion also. So ganglia blockers will also prevent releasing of epinephrine and norepinephrine from the adrenal medulla. All right. Next, as we're talking about cholinergic transmission, or postganglionic fibers that release acetylcholine, we have cholinergic agonists, plus acetylcholine esterase inhibitors, like an estigmine, like a physostigmine. We have antagonists, cholinergic antagonists, like atropine or glycopyrrolate, and we have acetylcholine esterase reactivators, like pralidoxim. As we're talking about postganglionic sympathetic fibers that release acetylcholine and are responsible for innervation of thermoregulatory sweat glands, these cholinergic agonists and antagonists also will work at this place, at this site. We see that alpha-2 receptors located presynaptically, and we have drugs that stimulate these alpha-2 adrenergic receptors like clonidine, methyldopa, and drug that blocks these receptors, yohimbine. On postsynaptic membrane we have alpha 1, alpha 2, beta 1, beta 2 receptors, so not alpha 2. Beta 1, beta 2 receptors, yeah. We have adrenergic agonists, like epinephrine, norepinephrine, again phenylephrine, isoproterenol, dopamine, dobutamine, and we have adrenergic antagonists. Adrenergic antagonist. Now we can see more information about botulinum toxin, and I suppose you should guess sites of action of botulinum toxin. Would like to remind you a little bit, yeah, as we discussed cholinergic synapse. We see it that botulinum toxin belongs to neurotoxin. It binds with specific proteins like a SNAP protein, yeah, or synaptosomal associated protein. It binds with vesicle associated membrane protein or VAMP, and also it interferes with syntaxin and can act on synaptobravin, and as a result it prevents releasing of acetylcholine in cholinergic synapses. Now let's see sites of action, yeah, based on this diagram, based on this illustration of transmission in ganglions or in parasympathetic nervous system and in sympathetic nervous system. What you can say where botulinum toxin will act. How do you think? You can type your answers. So, botulinum toxin. Will act first neuromuscular junction. Neuromuscular junction, because in neuromuscular junction we have cholinergic transmission. Second, botulinum toxin will act on autonomic ganglions. In ganglions of sympathetic nervous system and parasympathetic nervous system. Third, botulinum toxin will block postganglionic parasympathetic nervous system terminals. They also have cholinergic transmission. And fourth site of action of botulinum toxin is a postganglionic sympathetic nervous system terminals that release acetylcholine. The one set of action after poisoning with botulinum toxin is around 24 to 72 hours, and around 4 to 10 days as a peak of action. One set and peak. Recovering. Botulinum toxin produces irreversible inhibition in cholinergic synapses. as a result of these irreversible changes, recovery occurs by formation of new synaptic contacts. So basically, synapse should be formed de novo in new place in order to continue innervation, of organs or tissues. Another question: why botulinum toxin does not affect adrenergic synapses and it affects only cholinergic synapses? The answer is because cholinergic synapse has specific receptors to botulinum toxin. It has specific receptors, and botulinum toxin has two chains, heavy chain and light chain. As botulinum toxin binds to specific receptor, it appears inside after endocytosis, and light chain of botulinum toxin produces inhibition of mentioned proteins. Okay, that's it about general approaches of neurotransmission in parasympathetic and sympathetic nervous system and autonomic nervous system in general. And now let's talk about organs. Let's talk about receptors that located in these organs. And first, we should discuss eye pharmacology. Eye is a good point how to test your knowledges in pharmacology. And as we're talking about eye, we should say we should be focused on three processes. First, the regulation of iris diameter. Second, the accommodation. And third is the aqueous humor production. And first, let's discuss this regulation of iris diameter. Or pupil diameter, sorry. We have two types of muscles in the eye. This is the pupil, We have radial muscle, or muscle dilator pupilla, and we have circular muscle or muscle constrictor pupilla. These muscles have different receptors. Circular muscle has M3 receptors and radial muscle has alpha-1 receptors. And pupil diameter is more balanced between parasympathetic and sympathetic nervous system tone than action one of them. Now I will show you this on illustrations. Sympathetic nervous systems, the balance between them. So what happens if we will stimulate M3 receptors? We will get meiosis or pinpoint pupil. Why that happens? Because after stimulation of M3 receptors, we have contraction of circular muscle, and as a result, we will have a pinpoint pupil, and this causes meiosis. If we will block alpha-1 adrenergic receptors, we will also have meiosis. Why? Because in this situation, parasympathetic nervous system tone dominates. Why it is dominates? Because alpha adrenergic receptors are blocked and sympathetic nervous system unable to affect these receptors. So meiosis can be in two situations, yeah? In case of M3 stimulation or in case of alpha-1 blockade. And in case of M3 stimulation, we have like a cholinergic branch, yeah, affecting cholinergic branch, and in case of blockade of alpha adrenergic receptors, we have effects on adrenergic part. Stimulation of alpha-1 adrenergic receptors will cause mydriasis. Why mydriasis? Because stimulation of alpha-1 adrenergic receptors will cause contraction of radial muscle. Will cause contraction of radial muscle, and as a result, we will have mydriasis. Blockade of M3 receptors also will cause mydriasis, because tone of sympathetic nervous system dominates, as we blocked muscarinic receptors. So here we should remember definition, yeah, of antagonists, that antagonists just prevent binding of agonists. They will not produce effect, but they prevent binding of agonist to the receptors. So this is the first principle related to pupil diameter, regulation of pupil diameter. So once again, we have two types of receptors. We have adrenergic receptors and we have cholinergic receptors. The second principle I would like to mention to you is a regulation of accommodation. Regulation of accommodation. In case of accommodation, we have ciliary muscle. Let's draw a kind of basic state of ciliary muscle. This is a lens, and this is a ciliary muscle. And ciliary muscle has M3 receptors only. Stimulation of M3 receptors causes contraction of ciliary muscle. As a result, ciliary muscle becomes shorter, and lens becomes more spherical, because lens like a balloon. If you stretching in it, it's more flat. If you stop stretching in, it becomes more spherical. Yeah, so lens becomes more spherical. More spherical, and this causes spasm of accommodation. But in general, we have good vision for near objects, and we have bad vision for far objects. Blockade of M3 receptors causes relaxation of ciliary muscle. As we have relaxation of ciliary muscle becomes longer, and it pulling lens, and lens becomes more flat. As a result, this causes paralysis of accommodation or cycloplegia. We have bad vision for near objects, and we have good vision for far located objects. Is it important? Yes, it is important. Important for question solving. Yeah, especially in case of poisoning. Because you should be able to distinguish between cholinergic branch or adrenergic branch. And if you see in clinical vignette that patient has affected pupil diameter and accommodation, you should think about cholinergic part, yeah, M3 receptors, because only pupil and ciliary muscle has common receptors, M3 receptors. If you see affected only pupil diameter without acting on accommodation, then you should think about alpha 1 adrenergic receptors or about sympathetic nerve system. How this visual acuity can be mentioned, yeah, as an accommodation, as a visual acuity. It can be mentioned that patient has difficulties in reading newspaper, yeah, or difficulties in reading the books, yeah, the book reading. So this telling that patient has problems with vision for near objects, yeah, for near located objects. So once again, if we have, remember, yeah, if we have affected pupil diameter plus visual acuity, then we have affected cholinergic system. If we have pupil diameter only, then you should think about adrenergic transmission. Adrenergic transmission. Okay, and third part for today, most likely the last one that we will discuss, the aqueous humor production. Aqueous humor production. So let's draw eye as a cornea, and let's draw eyeball, iris, and lens. And lens. What we have? We have ciliary body here with beta 1, beta 2 adrenergic receptors. Also, alpha 2 adrenergic receptors are present. Alpha 2 receptors. So, this ciliary body produces aqueous humor, and aqueous humor going to this way into the anterior chamber and eliminated to the trabecular meshwork to the Schlemm channel. Schlemm channel. Around 90% of aqueous humor eliminated by this way, and 10% eliminated by scleral pathway. Scleral pathway. So, stimulation of beta 1 and beta 2 receptors. Why we're talking about beta 1, beta 2 receptors, not about one receptor type, but two of them, because equal amount of beta 1 receptors and beta 2 receptors are present in the ciliary body. So stimulation of beta 1, beta 2 receptors increases or enhances aqueous humor production. Blockade of beta 1 and beta 2 receptors decreases aqueous humor production. This is useful, in case of glaucoma, in case of treatment of glaucoma. So in case of glaucoma, we need either enhance elimination or decrease production. So beta blockers like timolol or betaxolol are useful because they decrease aqueous humor production. Stimulation of alpha 2 adrenergic receptors decreases aqueous humor production. And medication called apraclonidine. Did you remember clonidine and methyl-dopa? They stimulate alpha 2 adrenergic receptors, presynaptic adrenergic receptors. Topical agent available that called apraclonidine is similar to clonidine, but is available as eye drops. Also decreases aqueous humor production. And blockade of alpha 2 adrenergic receptors increases aqueous humor production. And medication, we said apraclonidine. Belongs to adrenergic agonist. So next, about production. This is how we can affect ciliary body. But we have a group of diuretic medications that called carbonic anhydrase inhibitors. let's say acetazolamide, and acetazolamide also decreases aqueous humor production. Also decreases aqueous humor production by inhibition of carbonic anhydrase. And diuretic that called osmotic diuretic mannitol. Also useful in glaucoma treatment. So once again, we should have balance between production and drainage. How we can affect production? The last group I would like to mention, so we said, yeah, it might be beta blockers, it might be a adrenergic agonist that called apraclonidine, carbonic anhydrase inhibitors, mannitol enhances elimination, and prostaglandin analogs. Prostaglandin analogs like latanoprost also enhances elimination by US scleral pathway. How we can affect elimination? Also, how we can affect elimination? First, we should open this Schlemm channel, and we should open trabecular meshwork that present there. How? We should cause meiosis in this patient. Yeah, why meiosis? Because in this situation we have contraction of muscle sphincter, yeah, and this causes opening of Schlemm channel. Meiosis enhances elimination. Drugs that cause meiosis: cholinergic agonists like pilocarpine, yeah. Adrenergic agonist. Could adrenergic agonist be useful in glaucoma treatment?